Okay, so let's continue then with neurocognitive methods in programming, although it's only neurocognitive methods in the winter term. So we now went over the first two bits of the course, which was this general overview, where I went over everything in not much detail, but so that you have heard a lot of things. And then we did this introduction to the MR imaging process with our magnetization vector and T1 and T2 time constants, transverse plane, and so on. But that's now also over. And now the next topic is, if you want, the biology of the whole process, which is called metabolism, hemodynamics, and the Bolt signal. If you read the Hüttel, as I requested, you will also find that we are basically just following the Hüttel in this regard. So the next topic after the MR physics and stuff is now the biology. And that's stuff that now again maybe from an applied perspective, so as a computational cognitive neuroscientist, I see these things essentially once a year in this class. Maybe not quite if we are talking about modeling the Bolt response, also in terms of a biologically plausible dynamic model. This I actually see more often, but the real kind of basic biological details, they are kind of, one knows them, that they are there, but one doesn't every day discuss the conversion of ATP into ADP or things like that in a cognitive neuroscience context. So just as a qualification of the stuff that we are doing, so it's important foundations. Some of them, if they enter the data analysis stream, they are not more important, but they are more visible than others. So the outline for the whole thing is to discuss first the idea of functional hyperemia, and then essentially establish this metabolic hypothesis of functional hyperemia, which essentially says that it's the metabolism that drives the hyperemia, so the increase in blood supply to a certain area. So that's kind of the first bit, and then to characterize a little bit the Bolt signal, because that's the basic signal that is of interest in cognitive neuroscience when you use fMRI. And finally, to discuss a little bit on what the precise neurocorrelates of the Bolt signal might be. Has anyone done something like this who had an fMRI course in their bachelor? An fMRI course? So the FU people, I guess. So you heard a little bit of this, I would assume. Okay, yeah, maybe. Yeah, judge for yourself how much you heard about this or not. I don't know. So who taught it, Jakob or? Okay. I don't know what he teaches. The first slide on functional hyperemia shows something discussed in the Hüttel that William James, one of the founding fathers of psychology, at least that's what I believe, because I didn't study psychology. William James apparently describes this measurement of cerebral blood flow volume increase by Angelo Mosso, and the idea is that there's a participant who lies down on this table. This is Angelo Mosso or Putin. I'm not quite sure who it is, but it's one of them. And this participant lays down on this table, and there's this cone that balances this table. And the idea of the story is that then Angelo Mosso gave the participant a cognitive task, and once so for example compute something, prove that where the Euler formulas come from or something, and then the participant thinks, and because this is kind of balanced and blood rushes to the head, it goes down on this side of the table. And then Hüttel discussed that most likely the story is not quite true. However, and this is the bigger point, and this is actually an important point, is that this idea that cognitive activity and blood flow or blood inflow, blood volume in the head are coupled is not something that came about with fMRI in 1992 or whenever fMRI as a field really started, but that was around for a long time, and that's I think an important message. So this whole thing didn't come out of the blue and has only been described since MRI is around, but it has been hypothesized and also experimentally shown actually previously. So this is another example for quite old experimental work that links an increase in blood flow and blood volume in the brain to not maybe perception, but at least external stimulation. So, so the experiment that is going on here is that I think it's dog, and the dog is stimulated at the sciatic nerve, and at the same time over the somatosensory cortex there is a measurement of pressure, so that's the oncograph. And what the experimental result is that if there is some stimulation to an external nerve, there is an increase in this oncograph, so an expansion of the brain tissue, and which they relate to the regulation of blood supply. So what they already, Roy and Schenken, published in 1890 is that these facts seem to indicate the existence of an automatic mechanism by which the blood supply of any part of the cellular tissue is varied in accordance with the activity of the chemical changes which underlie the functional action of that part, which also not only says that there's a link between or co-variation between stimulation of a peripheral nerve and brain supply, but also that this might be due to chemical changes that go along with activity changes that then induce this increase in blood flow. So what else do they write? Bearing in mind that strong evidence exists of localization of function in the brain, we are of the opinion that the automatic mechanism of the kind just referred to is well fitted to provide for local variation in the blood supply in accordance with the local variations of the functional activity. And this is also, of course, very important because the experimental finding is not that the whole brain increases in terms of its blood supply, but only on a local level, although this is not really shown here because this only shows where something is increasing. But this also means that this idea of brain mapping, so identifying areas of the brain that serve a certain cognitive function, or in this case, basically representing peripheral nerve activity, by means of local activations in blood supply has been around for a long time. Yeah? And this is not as esoteric as this here. So this is kind of a relatively straightforward experiment. So this was kind of a historical context to the whole thing. Now a little bit more formal. This term functional hyperemia, what is this supposed to mean? So it's supposed to mean the local increase in blood in response to a sensory, motor, or cognitive event. So some sensory stimulation, some motor output, or some purely mental cognitive manipulation might induce this local increase in blood. And I'm fairly unspecific what this in blood should mean, and we will see why that is later. So that's what functional hyperemia means. So functionelle Hyperemie, functional because it's related to a certain cognitive function, hyper because there's an increase, and emia because it's blood, so it's just referring to an increase in blood essentially. That's the functional hyperemia. The other question is then, how does functional hyperemia come about? And this is what also is a big topic in this session. It's that the main hypothesis how functional hyperemia comes about is a metabolic hypothesis, which basically states that local blood flow increased to meet the energy demands of neuroprocessing, so that the link is not, because it could also be different, right? So it could also be that neurons become active, and once a neuron becomes active, because it senses an action potential, this might just dilate directly a blood vessel, and then there would just be an increase, although and it has nothing to do with energy. It could be. So this is why it's at least it makes sense to give it a name and call it a hypothesis. Why is this kind of a sensible hypothesis? So the thing is that first of all, brain is quite metabolically expensive, and it eats glucose and oxygen. It only accounts for 2% of body weight, but uses 20% of the body's oxygen supply and 25% of the body's glucose, and contributes to 20% of the total basal metabolic rate. So it's actually really metabolically active, eats a lot of energy, and it does not store glycogen like muscles. So it needs to be constantly supplied with energy. Yeah? One can of course say that's a little bit stupid design. I think somebody this came up why it's not able to store glycogen and instead stores the glycogen in terms of belly fat. I think that's one of the ideas about what belly fat is about. So it's kind of the supply for the energy storage essentially for the brain. It's a little bit stupid design. I guess somebody will have an evolutionary theory why this is also positive, why that helps. But I don't know that. Do you know any evolutionary theory why it's good that the brain doesn't store energy?
No, I don't think it's good.
Okay.
There's no reason for it.
But you also don't believe in evolution, I guess, so as a biologist. Yeah, I don't know. I mean, it could also be that, one would have to follow that up, why that is the case. Maybe then the kind of expansion in terms of neural tissue was too fast to go along with a good adaptation of the energy demand. However, this is then a biological fact, and we have to now deal with it. So because it can't store energy, it makes sense that if it now increases energy demands, that it requires more energy, and that at least there is some mechanism that leads to an increase in the blood supply. So again, functional hyperemia, meaning increase in blood flow in a local area due to to some function, some neural processing, metabolic hypothesis, saying this happens because this area needs some energy, and energy needs to be supplied. So that's kind of the metabolic hypothesis. And to go more into the details of this, that's then the aim of the remaining slides, essentially. In the end, we will find out that it's not really clear why this happens, so that this is still an ongoing research field, which, however, that it's not quite clear why something happens, of course, doesn't stop anyone to use this covariation for other purposes. I mean, if you start thinking about things. Many things are not quite clear why they work, but the fact that they work doesn't stop anyone from using them. So let's go on. So then that was kind of definition of functional hyperemia and metabolic hypothesis. Questions about this right now? No. Then now to understand these ideas about the metabolic hypothesis in detail, it makes sense to review certain aspects of energy consumption and blood flow and so on, so that one understands certain terms and so on. So the next couple of slides will remind you of things that you have seen, I assume, many times. We will not go into much detail because I really assume that you have seen all of this. So, of course, you're all familiar with the basic anatomy of the neuron, with its soma and its axon. Of course, this is kind of an ideal of a neuron. If you look into neurons in different brain areas, for example, then they all look quite different. And if you look at neurons that relate to peripheral nerves and so on, they again might look quite different. But yeah, it's kind of the idea of a neuron. This is maybe something that shows things a little bit, or at least that shows them different. So it's a pyramidal cell in the cortex with some Golgi staining going on and some other cells here. There are numbers which your grandmother will ask you when, because you're in neuroscience, so how many neurons are in the brain? I mean, your grandmother is really great. But these are numbers that I never remember, because that's kind of the point. So the average-sized male brain has 86 billion neurons, 16 billion neurons are in cortex, 69, that's actually interesting, 69 billion neurons are in the cerebellum. So the vast majority of neurons in the brain are actually not in the cerebral cortex, but in the cerebellum. And there are 100 to 150 trillion synapses. So there are many more synapses than neurons, which in a way also means that the communication between neurons might be more important than the contribution of each individual. Yeah? I will not ask these numbers in the exam. But there are many. And the energy that they require, so basic energy that they require, so there are two processes that require energy, and one of them is the sodium-potassium pump. So you all learned about action potentials in school or in psychology or in college or whatever, and you know that the whole neural communication works because there are these gradients of certain ions over the neuronal membrane, so that there is, for example, a higher intercellular concentration of potassium and a higher extracellular concentration of sodium. And that these gradients of ions need to be actively maintained. If they are not actively maintained, essentially when you die, then everything breaks down. And to maintain these gradients, you learned about the sodium-potassium pump, which eats energy in terms of ATP, which is transferred or transformed to ADP. We will see a little bit more on ADP later. So that's one energy-consuming process. I think maybe even the bigger energy-consuming process, although I don't know whether I have numbers later on, but other very important energy-consuming process is actually the whole synapse business, which takes energy via various processes. So one is, of course, forming the vesicles that then get released once an action potential comes, and calcium gets into the presynaptic part, and these vesicles then basically fuse with the membrane, release the neurotransmitter, for example glutamate, and then there's stuff in the synaptic cleft. This needs to also be recycled, so this again requires energy, and maintaining the whole thing. So the synaptic activity definitely also requires a lot of activity in terms of setting up the whole process and also maintaining that it works. And to go more about the synaptic process and then also later understand a little bit more about this metabolic hypothesis of functional hyperemia, I'm also reminding you of some glia cells, in this case the astrocytes. For your who wants to be a millionaire questions, it might be worthwhile to also remember that there are other glia cells, namely oligodendrocytes, which provide the myelin sheaths in the central nervous system. How are the cells called that provide the myelin sheaths outside, so in the peripheral nervous system? Schwann cells, yes. So these are oligodendrocytes in the central nervous system, providing the myelin sheath, and microglia, which are immune cells. So, but we focus here on astrocytes. I just brought up them because this is kind of textbook undergrad knowledge, which is fun to revise. Now the astrocytes, actually, they have an important role in vascular coupling, which we will also then supplement with further details later on. They are also, and this is what this picture is supposed to show, they are kind of well suited to be kind of intermediary between neural activity and blood regulation, because they cover with their protoplasmic processes, so with their little feet essentially, they cover also small blood vessels, and as we will see in a second, they also can be located quite close to a synapse. Yes, and they are coupled to other astrocytes by gap junction, which means they can also quickly communicate and also make maybe more locally expanded effects. So this is what I actually wanted to mention. So the astrocytes can kind of enclose pre- and postsynaptic parts of the synapse. So the idea is here you have a dendritic spine, which I wanted to start actually here. So you have an axon terminal, which is the presynaptic cell. So you see some vesicles which can be released. You have a dendritic spine somewhere in the cortex, which forms the postsynaptic cell, and around it you have astrocyte processes. Yeah, so this is kind of a more idealistic representation. So here presynaptic neuron, postsynaptic neuron, and around it you have a astrocyte, which is located there to likely do things like taking up neurotransmitter that is released into the synaptic cleft and recycling that, and also then providing the synapse again with glutamate, and also kind of regulating the ion milieu and concentration of the minerals there. So together, these pre-postsynaptic and astrocyte units are called tripartite synapses. They are thought to contribute to the concentration of, like I said, ions, extracellular potassium, and do glutamate recycling. And then the idea is that they are sitting here at the synapses, so they actually they are sensitive to synaptic processing. For example, if in a local brain area there's now some activity and synapses are firing like wild, they have to do a lot of this glutamate uptake and regulating the extracellular potassium concentration. Now they can also make sure that that they regulate the blood flow to make sure that energy is coming, because they have their protoplasmic feet at the small vessels. Yeah, so that's this idea of a tripartite synapse. I think last year we figured that this is not kind of textbook knowledge in biology yet. Have you heard about this? The tripartite synapse? No. So it's not a concept that that is taught in neurobiology for biologists, which doesn't mean much. It just means that it's that novel that it hasn't entered the canon yet. Yeah, that's what it means. And maybe it will never. So maybe it's all wrong and we will never enter the canon. But then there's so many things in the canon that are also wrong, so it doesn't really matter. As long as the young people study something, it doesn't really matter what they study. So that, now talking about kind of the cells that are involved and also the synaptic processes. Another thing to remind you of, to make the story of the metabolic hypothesis then a little bit clearer, is this ATP versus ADP. I think most of you have seen that in your biochemistry courses. I would assume that ATP is just this molecule being composed of this adenine component, this ribose here, so this sugar, and then these phosphate units, if you want, and it's called triphosphate because it in this version it has 3 phosphate stuff, and these bonds between 2 phosphates, they are quite energy rich. So if this one of them is split off, there is energy being released. And to create ATP from ADP, this costs energy. So it's kind of the energy Bitcoin, so the energy currency of living things. Has anything changed in biology? Do they tell you it's ATP, ADP, and something?
Can be, no, not really. Sometimes GDP, but
no.
Maybe ADP.
Okay, so it also hasn't changed since I studied that, which was early 2000s. So it's like 16, 18 years ago. Okay. Now, to again understand what's going on with the metabolic hypothesis, it also helps to remind one a little bit about how ATP is actually generated, and especially from glucose and oxygen. That there's this process called glycolysis, which is the process of metaboling glucose into other compounds to produce ATP. And there are these 2 forms of aerobic glycolysis. It's especially interested if you do fitness or something like that, or sports, then it's all about aerobic and anaerobic. So the aerobic glycolysis is the one that actually uses oxygen. This is the one that actually yields a lot of ATP. So basically from one molecule of glucose, so sugar, and 2 oxygen molecules, you get 34 ATP molecules via essentially some basic glycolysis, and then the tricarboxylic acid cycle. You see that I'm pronouncing these words once a year. Following the TCA cycle, I guess, the mitochondrial electron transport chain. There is also anaerobic glycolysis, which is less efficient, but also quite quick, which converts glucose to lactate in the absence of oxygen. So if you run out of oxygen, you can still create energy from glucose, a little bit less efficient, and you get an increase of lactate, and then I think sportsmen or women are particularly interested, or at least there was a time when they were interested in their lactate measurements. I'm not that into current sports science, whether that's still that interesting, because you also have, like everywhere, you have things that are at one point very important and then they are less important at one point. But aerobic and anaerobic glycolysis exists to remind you of this, these basic processes. So this kind of the anaerobic way to process glucose to get certain intermediate compounds. And if you have oxygen in addition, then you can enter this TCA cycle, which some of you might have memorized in your undergrad studies. And from there, you get compounds that are then fed into this mitochondrial electron chain, which then can actually create quite a lot of ATP. So that's kind of this undergrad knowledge. Who has seen this in detail? So you have definitely seen that in detail. Has anyone else seen that in detail and learned the TCA cycle compounds? So what did you say?
The whole TCA and
but in what kind of subject? So what was
biochemistry.
Biochemistry. Yeah. You have an undergrad in biochemistry?
Toxicology.
Toxicology. Yeah. That makes sense. Yeah, these are the typical things. Okay, so this as a reminder, or maybe as something novel that you have never heard, then maybe have at least some look into it. Yeah, this is the slide that, or not the slide, but the idea that I was talking about earlier. So the question is how much of these processes that we talked about actually goes where, and the thing is that 75% of the energy budget are actually for signaling related processes. So this is kind of the restoration of ion concentration, resting potential generation, and transmitter recycling. So this is in a way all signaling related. So resting potential generation, of course, because you have action potentials, and transmitter recycling for in terms of the synaptic activity. Then 25% of the energy budget, this is about the role in brain, by the way, for non-signaling, for non-signaling processes like basically keeping up the infrastructure, the anatomical infrastructure and stuff. So what is shown here is postsynaptic potential, action potential, resting potentials, presynaptic and transmitter recycling. So this is all required for signaling. Housekeeping then meaning the non-signaling processes, like making sure that there's a cell, that the cell has a membrane and it has its different can do its things like expressing certain proteins by these processes from DNA to RNA to proteins and so on. Of course, this also takes energy, but the main energy that is required is for signaling. Good, so this was just a little bit, so these were some spots in energy consumption that are helpful when understanding the metabolic hypothesis of functional hyperemia, and things that you should know anyway, because it's good to have this kind of general knowledge. Next thing is more blood related, and this is more on the macroanatomy first of the cerebral cortex and the brain essentially, and I like to start from basics. So the basic thing is of course that humans, but of course also other mammals, have a vascular system which has two main compartments, kind of the arterial system and the venous system, and then you have of course the heart that makes sure that the blood circulates, and that also makes sure that the blood is reoxygenated in your lungs. And these are all names for arteries and for veins that are primarily interesting if you poke things into them in medicine. At that level, it's maybe not that interesting from a cognitive neuroscience perspective, because luckily we don't have to give any transmitter also, so we don't really need to remember the names of the arteries and veins, at least in the periphery. It makes some sense to know a little bit about them in the head, actually. So this shows the thing a little bit more idealized and a little bit more function-oriented. So remember that you have a heart, and that this heart has two chambers, and the right chamber taking basically the venous blood as collected in the vena cava and pumping that into the lungs. So you have here actually arteries, which are defined as blood vessels that carry blood away from the heart, although these arteries actually carry venous blood. So that doesn't really make sense. They carry deoxygenated or low oxygenated blood. So arteries not always have high oxygen saturated blood. These pulmonary arteries don't. They carry that to the lungs. In the lungs, you breathe in and make sure that via the capillary bed in the lungs, you have this exchange of oxygen and CO2. And then once it's run through there, it's collected again, and now oxygenated blood going to the left chamber of your heart and then being pumped via the aorta into the periphery. So this is oxygen-rich, CO2 poor blood, and the blue one is oxygen-poor, CO2-rich blood. What happens then is that, at least in the arterial system, you start out with these vessels that are quite muscular, so that have a lot of smooth muscle tissues, which gives them really like a firm structure. So if you ever worked on some corpses, not necessarily human, but also other animals, then you might have found that these arteries, they are like real things, so they're resistant a little bit to pressure and feel like real things. They, of course, split up and go to ever smaller things, and are then called, for example, arterioles, when they still have some smooth muscle cell. At one point, only the inner layer of these vessels is still maintained, and this inner layer is called endothelium, and this is then when they are in the when we are in the capillary bed. And then in the venous system, we have this endothelium, and all these veins, they then that collected here. You also have these valves in the veins, which sometimes go wrong. They are less nice kind of anatomical structures, so they don't really feel like something proper. So because of the muscle here, but of course the pressure in the system is of course a lot higher, so you need much better and tighter tubes essentially to hold that. Yeah. So remember that that there are different anatomical components. So going from large arteries, which have a lot of smooth muscle and can actually actively constrict, to small arterioles, which still can be constricted by smooth muscle cells, and then to the capillaries, where actually the O2 is extracted, and also the glucose is extracted, and CO2 can be uptaken. They can also be constricted, but not by themselves, because they don't have smooth muscle cells anymore, but by pericytes, so cells that regulate that. And then from the capillaries, you can go, especially in the brain, via small veins, and then what are so-called sinuses, so long venous channels that are actually formed by the meninges, so the skin of the brain, and from the primary draining system to then veins proper. But they are mainly passive connective tissue. Yeah? Why is this interesting for cognitive neuroscience? because the question is, how is this regulated? So where are parts that can regulate actually blood flow? And it's obviously a bit more intuitive to think that regulation happens at this arteriole level or capillary level. And then, of course, the question is in terms of the spatial specificity. Now, basically how close can two things be in the brain so that we can still distinguish them based on their blood supply. That of course depends on where the regulation happens, right? So if the regulation would happen at the level of the aorta, then the whole brain would always get more supply or less supply, so it would not be possible to do some functional mapping. Obviously, you know that it's not the case. Of course, there is some regulation of blood supply, of course, at the level of the aorta, but that's more general, central. But it's more this regulation that happens here that we're interested in, and this is what we will see also in a second. This is now just reminding you of the blood supply of the brain, which you should actually know about. So the is of course the aorta, and then leading up to a subclavian artery, and then there are two main supply chains, one being the carotids, which you can feel at your neck, and there's also the vertebral artery, which goes basically up your back neck, how that's called, and enters then at the base of so it goes in via the foramen magnum. No, there's some holes, I think, in the lower surface of the skull where it gets through. And then there are three main arteries that basically supply the brain, and they are actually relatively easy to remember because it's one anterior, it's a medial, and it's a posterior. Here you see kind of the vertebral areas going up here, and then supplying mainly the posterior cerebral artery. Is it shown here? I don't think it's shown here. There will be another picture that's shown better. And the carotid artery is mainly supplying the media and the anterior part. So that's where the blood comes from. Yeah, here it's shown in a different way. I think I have three pictures of this. So in terms of the arteries, what you see is here the back is essentially supplied by the posterior. The anterior, and that's kind of important, mainly supplies the medial part of the brain, and this stuff where the motor function sits and language and so on, this is mainly supplied by the middle cerebral artery. So that goes from essentially, you see it here? Not really. So it goes comes in at the base of the brain in the middle, and then it goes outside. And so if you have infarctions with the media or middle cerebral and artery, that's usually the worst, because that's really supplies a lot of fairly important functions. In terms of the venous system, you see here that there are these, especially there where the two hemispheres meet, there are these large sinuses, and yeah, they're called sinuses. There are other sinuses at the skull, which are these holes in your skull. They are also called sinuses. Sinusitis, if you have a cold, some people have sinusitis. That's not these sinuses. That's the holes in your bone sinuses. And the whole thing is, of course, not the sign, it's the sinus. Anyway, so this is another representation of how that works with the brain. So the anterior doing the medial part, so you see a little bit of yellow here, up here, but if you look at the brain from the side, you don't see much. The medial cerebral artery, you see quite a lot. The posterior cerebral artery, you see only a little bit, and the rest of everything that you see, especially this black stuff, that's supposed to be veins. Yeah, So this is kind of the rough macroscopic supply of the brain. You see these things actually mainly when you see what you see of this if you do fMRI studies is that you get these bright spots in your images, which are then the pictures of the arteries or the veins in the big when they are still big. So you, for example, you can quite usually you can see this circle of Willis here. So where all these connect, you see, for example, these things, you see them in pictures, in fMRI pictures. It makes sense to look at them, and yeah, then you know what they are. But these smaller things you actually don't see in fMRI or T1 images. And this is kind of the small level that the whole thing then gets to. So this is the capillary bed, and you see. So what happened here is that I think it's using some chemical, is brain perfused, and then the whole brain tissue is extracted, and the only thing that remains is the capillary bed. And this is what's shown here. So you see that it's really densely supplied with capillaries. I'm looking for a break point here. So let's briefly do the remaining things. So these are, and this is again talking about the spatial specificity. So we are now talking about the brains, and of course about the brain, and of course the arteries get smaller and smaller. The question is, if the control of blood supply is happening, for example, at the level of the distributing arteries, which have a diameter of 150 to 200 micron, these supply 3.5 times 2 millimeter of cortex. So this is the largest specificity in terms of anatomical specificity for functional activation that one can get. If it's happening at the precortical arteries, then it's going to smaller levels. And this is all still at the outside of the cortex. If it then these intracortical arteries, they take right angles and go into the cortex, they then supply even smaller aspects. Yeah. So it's not that one in the end we can say it's at this level or this level or this level that there's a kind of a clear-cut and easy answer. But it's something one should keep in mind when talking about and interpreting also fMRI pictures, because it's not, so this is essentially when I said medicine, what I took from the whole thing is, first of all, everything is adjusted in a way that it works, so things seem to be optimal quite often. And the other thing is that it's all quite complex, so it's not simple. Nothing is kind of like, if you would design it, you would make it much simpler because it's easier to understand in terms of these things and where things are regulated. I guess you can find experimental evidence for each of those being important for regulation of things. So this shows here the Calgren sulcus, so the visual cortex, and you see how from the surface of the brain there are some arteries going in. So these are these intracortical arteries, and then you still see a lot of capillaries and high. And what this also should stress, I think that's what's discussed in the video, is that especially where you have high density of cell bodies, so in layer 4 of the visual system, you have a lot of vascular ramifications, so that there's a covariation between there are many cells and then there are also many blood vessels, very small blood vessels, capillaries. Good, so let's take a break here and then discuss cerebral blood flow and remaining things in the next hour.